appeared to be normal. What you can see is they look a lot like the severe head injury. So it may not be the severe head injury, it may be the common mild head injury that's important. So proposed mechanisms for this. I talked about the alpha-synuclein protein that accumulates in those Lewy bodies in brain neurons. Head injury in animal models increases levels of that protein in the brain. And in humans who've had severe head injuries and died, we can also see increases in that alpha-synuclein protein. So that may be the mechanism of action of head injury. Now, pesticides. Many of you are probably familiar with this association. This has been observed worldwide at least 40 studies. And a pesticide, I should define, is a broad range of compounds. Insecticides, herbicides to kill plants, fungicides for fungus, rodenticides, pesticides, which kill fish, and acaricides, which kill worms and parasites. Now, these are broad classes of chemical compounds, and the problem with most of these studies is that these compounds chemically may be very dissimilar, and very few epidemiologic studies have actually studied specific compounds. So we set out to do that. We studied a very large group of 84,000 pesticide applicators in Iowa and North Carolina. What's key about these folks is that they know what they were exposed to. Most people, we don't know. We don't know what's out there. We don't know what's on our fruit. These folks know because they're professionals. They buy it. They apply it. We went out and we identified every person with Parkinson's disease in this cohort of 84,000, and we found 110 people with Parkinson's. We matched them to 350 people without Parkinson's, or controls, and we asked them about 31 pesticides that we thought were suspicious and might be related to Parkinson's. Of those 31, only 2 were associated with Parkinson's: Rotenone and Paraquat. Both of those increased risk 2 to 3-fold. What's really interesting about Paraquat in particular, it looks almost identical to MPTP. This is Paraquat, the molecular structure. This is the molecular structure of MPTP. That's the compound that was synthesized and caused acute Parkinsonism. Rotenone works by killing mitochondria. Works by the same route that MPTP does. So we found these two, we asked them about 31 pesticides and only 2 of them popped out. And they are both compounds that either look like or act like MPTP. We've studied other pesticides, and many of you may have heard of 2,4-D. It's an herbicide. It's been used since the 40s. It's used on golf courses. It's a component of Agent Orange. Agent Orange is primarily composed of 2 compounds, 2,4-D and 2,4,5-T. There is also dioxin in there. Many people in this room were probably exposed if they were in Vietnam. And a couple years ago, the Veterans Administration determined that exposure to Agent Orange was a contributing factor to Parkinson's disease and was a reimbursable service connection. Now, I've talked mostly about occupational pesticide exposure, but of course this is the tip of the iceberg. We're all exposed to compounds throughout the environment. We don't know which compounds we're being exposed to or how much. A study by our colleagues down in Southern California looked at well water and found that people who drew their well water from areas where Paraquat was being used around orchards were more likely to have Parkinson's. Solvents. have been looked at in Parkinson's disease, though not very much. A solvent is a substance, it's a very broad definition, that can dissolve another substance. They're everywhere. Fuels, paints, glues, lubricants. There have been a few studies that found associations with solvents, but not very many. And the problem, again, as for pesticides, it's a very broad range of substances. This changed in 2008. In Kentucky, some investigators noticed that several of their patients had all worked in the same small factory. And they'd all done the same job, which was degreasing of metal parts using a compound called TCE, or trichloroethylene. They went out and they tracked down their coworkers who had worked at the plant at the same time. They found 25 of them and they examined them. They had mild signs of Parkinsonism. So we saw this and we decided that we needed to investigate it further. So trichloroethylene has been around for a long time. Since the 1920s. It's used primarily now to degrease metal parts in the automotive industry and other similar industries. It was used in the U.S. for many years to clean computer circuitry. It was the predominant dry cleaning solvent for many, many years. Until the 70s, this is amazing, it was used as a surgical anesthetic and primarily as an obstetrical anesthetic. And it was used to decaffeinate coffee. It's still around. It's in typewriter correction fluids, no brand names. Adhesives, paints, carpet and spot removers. It's being phased out, but it's still being used extensively, 10 million pounds annually. It's the most frequently reported contaminant in groundwater. This map down here shows the density of Superfund sites that have trichloroethylene in them. Let's see here. Virginia, my geography, there we are, is one of the intermediate concentration sites. And what happens with TCE is it gets into the soil. In the air, it goes away. It evaporates, it's gone, it breaks down. But in the soil, it gets into the groundwater, and it stays there. And it can stay there for years or decades. Once it's down there, it distributes. This is actually from Dayton, Ohio, where there was a factory that used TCE, and it got into the water system, spread out underneath the town. And they're called plumes. There are plumes of trichloroethylene all over the country. It's found in foods. It's found in human breast milk. So TCE is out there. And so we studied it. Back to our twins. These are what we call our job-specific interview modules. So in order to determine if somebody was exposed to a compound like TCE, you've got to take a really detailed history. So we have about 50 of these where we ask specific questions depending on what job you had during your life. And some of these poor folks, these twins, we asked them 40 of these. And the interviews take 5-6 hours, and they're terrific to participate. So we looked at aircraft mechanics, carpenters, and all these industries. And we found that there were 3 solvents that were associated with Parkinson's risk. Carbon tetrachloride doubled risk. Trichloroethylene increased risk 6-fold. And perchloroethylene, which is another solvent that's used in dry cleaning, increased risk 10-fold. How might they work? This is one of the metabolic intermediates of trichloroethylene and perc in the body. It gets transformed into this compound. MPTP's on the left, Paraquat's in the middle, TCE and perc is on the right. It looks like MPTP. It acts like MPTP. It's toxic to mitochondria. And in rat models, it increases alpha-synuclein protein, and it damages very specifically that part of the brain that's affected in Parkinson's, the substantia nigra. So we're very concerned about TCE and hope to be doing some more research in that area. Finally, one other study that we have ongoing. We're looking to see if PCBs, polychlorinated biphenyls, might be related to Parkinson's. All of these compounds that I've been talking about. They are very persistent in the environment. That was their attraction to industry, is that they're not going away. But that means that even after their use has been discontinued, they persist. They persist in the environment, and they persist in our tissues, especially in fatty tissues where they can be, all of us have measurable levels of PCBs, for example. So this is a study that's funded by the Department of Defense. We're looking in Alaskan natives, Eskimo populations. They eat sea mammals, walrus, whale, and those are at the top of the food chain, and they concentrate PCBs. And Alaskans have very high levels of these compounds, so we're conducting a study up there right now to look to see if the rates of Parkinson's disease are higher among native Alaskans. So I've mentioned a lot of environmental risk factors for Parkinson's, but the vast majority of people who are exposed to these compounds don't get Parkinson's disease. Why might that be? Well, virtually all common disorders are not caused just by genes or just by environment, but by combinations. So as an example, heart disease. We all know that our environment affects our risk of heart disease. Smoking, pollution, high-fat diet, stressful lifestyle, and also by genetics. There are people who just cannot metabolize fats. They have much higher levels of cholesterol in their blood regardless of their diet. Diabetes is partially genetically determined, and all of these factors act together to increase risk of heart disease. So here's an example. These little pie diagrams. It can be confusing, but I'll try to walk through it. It confuses me, probably not you. So our disease that we're using as an example is a broken wrist. A broken wrist is caused by a variety of factors. Clearly, the factors that we're looking at are stairs, ice, osteoporosis, thinning of the bones, porch light burned out, high heels, and alcohol. If you only have stairs and ice, you do not break your wrist. This pie has to be completed for this person to break their wrist. If you have 4 of these, stairs, ice, osteoporosis, porch light, high heels, alcohol, any combination, you complete the pie, that person breaks their wrist. We think that's similar for Parkinson's disease. Now there's another situation where factors don't just add up. Factors increase the potency of another factor. So in this example, we have an environmental factor over here. Looks like my E fell off. And we have a gene here. This person does not get PD. Over on the right, we have someone who has the same environmental exposure. They have a different gene. They complete the pie. They get Parkinson's disease. This is what we mean when we say gene-environment interaction. The effect of the environment is different in these two people depending on which gene they have. The genetic variant modifies the effect of the environment. So here's an example of that. This is asbestos, smoking, and lung cancer. If you have neither, your risk is normal. If you have, I changed this. So it's a gene and smoking and lung cancer. So if you have just the gene, your risk of Parkinson's, your risk of lung cancer is increased 3-fold. If you just smoke, your risk of lung cancer is 10-fold. If you both smoke and have the gene, your risk is increased almost 20-fold. So this 6 here, this amount is the interaction. It's the additional effect that they have together. So I'm going to give some examples of what we have observed in Parkinson's disease for gene-environment interaction. Thank you. So many of you are still awake. This is a paper that we studied, that we published earlier this year. Now, the gene I'm going to talk about is the gene that makes alpha-synuclein protein. And the environmental factor in this case that we looked at is head injury. Now, I put up some of the head injury data earlier, but what I didn't show is that not all studies found an increased risk of Parkinson's disease associated with head injury. To the right, they found an increased risk. To the left, they actually found a decreased risk. Why might that be? Why might there be inconsistency between studies? Well, we think it might be gene-environment interaction. So in this graph, we're looking at the effect just of having a particular genetic variant in the alpha-synuclein gene. On the left, they have the normal gene. On the right, someone has the genetic variant of alpha-synuclein that makes more alpha-synuclein protein. Their risk is increased 50% compared to the person who doesn't have this genetic variant. So we ask the question, does having this variant modify the effect of head injury? And this is what we found. This is the increased risk if you have the variant in the alpha-synuclein gene. Increases risk about 50%. This is the effect of head injury alone. If you don't have the genetic variant and you get the head injury, it increases your risk about 70%. This is the combined effect. It increases risk 1,000%. This section right here is gene-environment interaction. This is one of the largest risk associations ever found in Parkinson's disease, and we think this is the tip of the iceberg. Another example of gene-environment interaction in Parkinson's disease, this was published 2 days ago. We're looking at a gene. In this case, it's called GSTT1. The name's not important, but it is an enzyme that metabolizes environmental toxins. So we thought maybe it would have a relationship with Paraquat because we've observed that Paraquat increases risk. So we thought perhaps a gene that metabolizes toxins might interact with exposure to Paraquat. And this is what we found. If you have the defect in the gene, it increases your risk about 40%. If you are exposed to Paraquat, it increases your risk 100%. It doubles it. If you have both, it increases your risk 1,100%. This red part is the interaction part between the defective gene and the Paraquat. So, is Parkinson's disease due to a combination of environment and genetics? Yes. Parkinson's is a complex disease. Like all complex diseases, every case of Parkinson's disease is different. Every one of those pies, that's a combination of a variety of risk factors. Every individual has a different combination of those risk factors. They probably work through similar mechanisms. They're probably toxic to mitochondria. They probably increase levels of proteins such as alpha-synuclein that bunch up together, clog up neurons, and ultimately lead to Parkinson's disease. So this is just an example of common factors that might all act together. So over here we have some environmental factors, rotenone, MPTP, Paraquat. This is actually the TCE, the solvent. We have genetic factors, so variants in a variety of genes. The genes, the vast majority of the genes that are linked to Parkinson's disease don't cause Parkinson's disease. They increase the risk 10, 20, 30%. The genes that cause Parkinson's are very rare. So we think that the genetic factors that are involved in causing the vast majority of Parkinson's disease are those that interact with the environmental factors. And together, as I showed in the gene environment slides, they interact perhaps on the same mechanism, perhaps on mitochondria, and ultimately lead to what we recognize as Parkinson's disease. So next steps. We need to continue to look for these interactions. We need to understand the epidemiology of Parkinson's disease. We don't know how many people have it. In California, we have something that's unique in the country. It's called a disease registry. There's a state law that was signed into law in 2004 by Governor Schwarzenegger. It is still funny. He signed this bill into law, but the stipulation was that the state would not spend $1 to implement it. So it's a law, but 99% of the doctors in California do not know that it's a law. They are legally required to report all cases of Parkinson's disease, but they don't. But we got some funding from the Department of Defense several years ago to actually go out in several counties in California and try to identify every single person with Parkinson's disease. So we've gone into every neurologist's office, internist's office, hospitals, and we've flipped through their charts. We are deputized agents of the state of California, and we've tried to identify every person with Parkinson's disease in our county, in Santa Clara County. And what we found is nearly double the rate of Parkinson's disease that we expected to find. We expected to find about 2,500 cases, and we found over 4,000. So we don't know how many people have Parkinson's disease. What we're trying to do now with this information that we know all 4,000 people with Parkinson's disease is we're going to link with what's called geographic mapping. We're going to link where they live with the frequency of Parkinson's, and we're going to try to find if the areas that they live are in clusters, and if so, if we can find what's different about those areas. Were pesticides used there? What's their source of water? So this is a great